hopefully you'll find this as helpful as you've found the respiratory and digestive videos. So first, you can follow along in your lab manuals on page 263. We're first going to talk about some of the gross anatomy of the urinary system, and then we'll go into sort of the microanatomy. We'll take a look at some of these structures in complete anatomy. We'll talk about some of the flow and the physiology behind the urinary system, and we'll tie it all together with a really cool 3D video before we make our way to histology. So the urinary system has an incredibly important job, several jobs in fact, but one of the most important being maintaining blood volume and composition, right? And the structural and functional unit of the kidney is going to be a nephron. So we'll talk about what all that means in just a second. So I want you to make sure that you know where the kidneys are relative to other structures in your body. And the kidneys are located along the posterior body wall from the vertebral levels T12 to L3, with the right kidney slightly lower than the left kidney because of the location of the liver. The kidneys are retroperitoneal structures, and I think this cross-section does a good job of showing us, with this being the anterior view and this being the posterior view, it does a great job of showing us that some of the structures that we saw in the digestive system are enclosed in this blue portion within the peritoneum, right? And so we call that intraperitoneal. So intra meaning within. But our kidneys are going to be behind. So retro meaning behind, retroperitoneal. And so we're first going to focus on the kidneys, but then when we look at the rest of the urinary tract, I want you to keep in mind that the ureters, as well as the urinary bladder, are all retroperitoneal. So let's talk about some of the structures, some of the gross structures of the kidney. And so the outermost structure that is going to be visible on most classroom models or when viewing an actual kidney is likely going to be your fibrous covering, which is going to be your renal capsule. And this renal capsule is going to be composed of dense irregular connective tissue. And then you will have two major regions of the kidney. You will have an outer renal cortex and an inner renal medulla. And so these are the names for the regions of the kidney. Now these regions are going to have quite distinct features in each. So the cortex is going to contain your corpuscles, your proximal tubules, distal tubules, and paratubular capillaries, whereas the medulla is going to contain your nephron loops, collecting ducts, and your vasa recta. But one thing I want to sort of caution you about is make sure that on your quizzes and exams, you're specifying, if you're asked about a region or structure, if it's something that you can find in multiple organs that we've discussed this semester, make sure you define it with a prefix. So, for example, we've seen quite a few organs at this point this semester that have a capsule and have an outer cortex and an inner medulla. So you have to make sure that you're specifying by putting the prefix renal. So within your medulla, you will have medullary pyramids. And those are going to be those cone-shaped structures that we'll see in a cross-section of the kidney in just a second. And between each pyramid, we're going to see renal, or you can call them the renal pyramids. Actually, on your quiz and test, it'll be best if you put renal pyramids. But between each pyramid, you will find the renal column. And so this is just a general term for what lies between each renal pyramid. Okay? So then the renal medulla will collect the urine that gets made in the pathway that we'll take a look at in just a second. But it will collect them in structures called papilla, renal papilla, and then they will collect the urine and route it to minor and major calyces, which will then route the urine to the renal pelvis. Which will be within the renal sinus, which is that area where you'll see the renal pelvis feed urine into the left and right ureters. And where you see the ureters exit the renal artery enter and the renal vein enter, you will see an indentation that will be your hilum of the kidney, right? So this is going to be a pretty good indication, and you'll be told whether you're looking at an anterior view or a posterior view, most likely. You can look at the indentation to try to figure out if there's no other structures around it. The indentation will kind of give you an idea which kidney you're looking at, whether you're looking at the left or the right. So what's going to determine whether we are looking at a minor calyx? So calyx, C-A-L-Y-X for the singular version. is 2 or more of these, so usually 2 to 3 minor calyces, will drain into a major calyx. And usually a major calyx, 2 or 3 major calyces, will then drain into the renal pelvis. So if it seems like it's directly connected to the renal pelvis, then it's most likely going to be a major calyx. So you can drain urine straight from a renal pyramid to a major calyx, and you can skip the minor calyx. So the key there will be to try to look at whether it goes directly to the renal pelvis or whether it merges, whether 2 or 3 minor calyces merges into a major calyx before it drains into the renal pelvis. So let's take a look at what that looks like. So here we are in an artist depiction of a cross-section of a kidney. So again, the renal hilum is going to be that indention in the kidney where you will see the renal artery come in from the abdominal aorta and the renal vein exit from the kidney to the inferior vena cava, and where you will also see the ureters exit, taking the urine from the renal pelvis. So this area is the pelvis right here. Okay, and so this is going to be our medullary pyramid. This right here, this little cup-like portion being the papilla that's going to collect the urine. This, because we're going to take 2 of these minor calyces and we're going to merge them into this portion right here, these will be minor calyces and this will be a major calyx. This rectangle I'm drawing here, this region will be a major calyx because they're going to merge before they get to this pelvis, renal pelvis region. Whereas something like this goes directly to the pelvis, so this portion could be considered just a straight major calyx. And so we're going to take a closer look at the microanatomy in just a second, but just so you keep in mind when we zoom in on what's happening, the papilla, the cup-like portion here, the minor calyx, which is collecting urine here, and all of this portion of the collecting duct will be in the renal pyramid of the medulla. And in the cortex region, we're going to see a majority of the parts of our nephron. And so this is a nice picture of a kidney dissection where you can see, so there is our medullary pyramid. There's another one. So that means the outer covering is going to be our capsule. This portion right here, this region, is going to be our cortex, and everything inward is from the cortex to the calyces is going to be our medulla. And so again, to decide whether or not it's a minor calyx or a major calyx, we'll check to see if it drains into a sort of merge point before we get to the pelvis. And you can tell which one the pelvis is because it's the portion right before the ureter. So find your ureter. That's going to be collecting urine from the pelvis. So if we work backwards, and so then that will make this the major calyx. And the fact that it's collecting urine from multiple minor calyces and multiple medullary pyramids is going to tell you that it's safe to call this a minor calyx and major calyx. Okay, so you can take a look at this picture to practice. And this would be a good time to pause and try this on your own. And now let's review. So this is dyed with a colored dye to kind of show you a little bit more easily what we're looking at here. And so you can really make out a distinct portion. And this is going to be the region we call the cortex, right? And you can really make out your medullary, your renal pyramids quite clearly here. And so when we're trying to decide whether something is a minor calyx or major calyx, we'll first identify where our ureter is exiting, which means the portion right before that, past the hilum, will be our renal pelvis. And so, for example, I think you can kind of see more clearly over here. We've got two medullary pyramids. And two of these structures that are going to collect urine into this merge point before it gets to the pelvis. And so this would be our major calyx, and each of these little branch points would be a minor calyx. Okay. So now let's get into some of the microanatomy of the kidney. And I want to start with taking a look at the structural and functional unit of the kidney. And the first part of that is going to be the first part of our nephron is going to be our renal corpuscle. Okay, and so the corpuscle is actually going to include not just the glomerular capillaries that you see here, but it's also going to include the capsular space as well as the parietal layer of this capsule. And this is going to be our sort of MVP. It's going to be our primary location for filtration, which is going to be one of the three main functions of our functional unit. And we're going to have two types of these nephrons. Our cortical nephron, which is going to be, like the name says, in the cortex, and our juxtamedullary nephron, which, if you think about, if any of you in your English classes talked about juxtaposition in your writing, juxta means near or next to, so it'll be near your medullary region. And the parts of the nephron we're going to look at will include our renal corpuscle, which is going to include our glomerulus and our glomerular capsule and our capsular space. And then we'll have our let's back up a bit. Jumping ahead of myself. And our tubules. So that'll include our proximal tubule and our nephron loop and our distal tubule. Okay, so notice how I didn't put anything else beyond the distal tubule. So these are the only parts of a nephron. So if you get asked at any point, what are the parts of the nephron, make sure you don't include the duct system. Okay, the parts of the duct. Which is the part of the collecting system. Very, very different, very, very distinct. Okay. So, here are the three parts of our glomerular capsule. So, our parietal layer of the glomerular capsule is going to be made of simple squamous epithelium that is not permeable. Then we'll have our capsular space. And this is going to be our visceral layer of the glomerular capsule, which is going to be pretty much the same thing, which is going to be made up of modified epithelium called podocytes. And these podocytes, the pedicles of these pedicles, I'm not sure how to pronounce it, are going to form these little filtration slits around the capillary and the capillaries, which is going to be sort of like your coffee filter in a way, as the blood filters several things that will leave the glomerular capillaries and enter the capsular space. And so on one end, we'll have the blood entering the glomerular capillaries through the afferent arteriole, and then being filtered through the glomerular capillaries and then leaving through the efferent, right? So A before E, so coming in through the afferent, out the efferent. So this, because one side of it is going to be where the blood vessels come in, this is going to be our vascular pole, whereas the other side will be our urinary pole, because that's where the filtrate is going to leave the capsular space and enter the proximal tubule. So this is a nice picture of a scanning electron microscope photomicrograph of these podocytes, which again are these modified epithelial cells that are going to be what makes up the visceral layer. They are one and the same. They are the visceral layer of the glomerular capsule. And these that you see here are their little extensions, their little legs extending. And between these legs, you can see it creates these filtration slits through which solutes and electrolytes and small things can exit into the capsular space. So I think that looks super cool. Okay, and this is just a picture from your textbook showing you the difference between cortical and juxtamedullary nephrons. So as you see here, cortical nephrons are certainly going to be, a majority of it will be in your cortex. You may have a little bit of the nephron loop sort of dip past the border, but a majority of a cortical nephron will be in your cortex, whereas your juxtamedullary nephron, the vast majority of your nephron loop will dip deep into the medullary region. But even with juxtamedullary nephrons, you're still going to have your renal corpuscle, and your proximal tubule and your distal tubules still in the cortex. And if we're talking about a cortical nephron, we're talking about our peritubular capillaries, and when we're looking at juxtamedullary nephrons, they will also have a portion of the capillary beds surrounding the nephron loop, called the vasa recta. And so notice how there's like a second capillary bed with these. Okay, so let's walk through the blood flow together. And it'll kind of make a little bit more sense once we nail the blood flow down. So first, as we take the freshly oxygenated blood that left our left ventricle through the aortic arch, then down the descending thoracic aorta, past the diaphragm, down the descending abdominal aorta, then our oxygenated blood is going to feed this blood into the left and right renal arteries. And then we'll get to the left and right segmental arteries. And so what's happening here is as we're coming in through the renal arteries. And after you pass the hilum, you're going to branch. And remember, just like we looked at branch points for the vessels, learning our blood vessels, you want to look at the branch point, because the point where you see your renal artery, the segment between the renal artery and your next branch point is going to be your segmental artery. So it's a fairly short portion relative to your other vessels. And then we So I'll for a moment, I'm going to stop writing left and right because let's just assume that we're just looking at one uh, kidney at a time and zooming in. Um, your segmental arteries will then feed blood into your interlobar arteries. Now, each of these regions with a single renal pyramid, so just like how we talked about lobes with the lung, we also have different lobes of the kidney. And so when we're going between the lobes here, as we go between the lobes, we're going to go interlobar because I want you to think lobes. Okay, so we go interlobar, and then we're going to sort of loop around the pyramid like so. So there's interlobar. And then we'll see our arcuate arteries. And arc kind of reminds me of archery. I used to participate in the Yellow Jacket archery team at Georgia Tech. And it was one of my favorite ways to relieve some stress. And that's what I think of quite fondly when I see this, what sort of looks like a crossbow right here. And it sort of arcs around the renal pyramid. So then from the arcuate arteries, so if this is a lobe, if this huge portion is the lobe, remember what we do with cutesy things, so baby versions, tiny versions of things. Above each lobe, the cortex can also be divided into little lobules on top. And so we will go between each lobule, ULE, for cutesy small things, interlobular arteries. Okay, so we're going between our lobules. And we're going to get smaller and smaller. Then when we zoom in on one of these lobules, you'll see your renal corpuscle. And what's really cool about this is that this is one of the few places in your body where an arteriole will feed, will go into a capillary bed and then will exit through another arteriole, right? So very, very special. So from the interlobular arteries, we'll go to the afferent arterioles. Then we'll get to our glomerular capillaries, our capillary bed, where again is going to be our primary site of filtration. And then we'll exit there through the efferent arterioles. So if we were to only look at one at a time, So let's say on just one side and talk about all of these as singular, because spelling is going to be quite important on your quizzes and tests. We'll say renal artery to the segmental artery, to the interlobar artery, to the arcuate artery, to the interlobular artery, to the afferent arteriole, to the glomerular capillary bed, to the efferent arteriole, and just to show that we're approaching another capillary bed in a different color, we'll go to the peritubular capillary bed. Okay. And so peri meaning near or around, right? And then this peritubular capillary bed is going to then collect deoxygenated blood from the surroundings and take it to the, we're going to just go back exactly the way we came. So we'll get to the interlobular vein, then we'll get to the arcuate vein, then we'll go back between the lobes, and eventually drain from multiple interlobar veins to the renal vein, which will leave that hilum and drain into our inferior vena cava. Okay, so this blood flow is incredibly important as we look at what's going on in the kidney. And one thing that's very important to point out is you can use your arcuate artery and your arcuate vein as sort of like the border that's going to divide your renal medulla from your renal cortex. Okay. And so let's look at some of these structures in complete anatomy. Okay, so I'd actually like to start here and then I'll zoom back out from a kidney lobe to show you some of the other gross anatomical structures, but I really, really love this model that they have in complete anatomy of the kidney lobe. So here we are with our renal papilla that's collecting urine from these various collecting ducts in our renal pyramid. And so if we follow the blood flow, so this is going to be, since we're going between a lobe, right? If we imagine another lobe right here, this will be our interlobar artery, which will then go to the arcuate artery, which will then go to the interlobular. So if I zoom in here, we can see that we're heading to the interlobular artery. We'll come into the corpuscle through the afferent arteriole, go through our glomerular capillaries, then we'll exit through the efferent arteriole. We'll come back out. And we'll loop around here to our peritubular capillaries. There's that gas exchange. Then we'll exit the peritubular capillaries and head through the interlobular vein, down the arcuate vein, then back down the interlobar vein. Okay, and this is also really cool because you can kind of see the difference between the cortical nephron right here versus something that's very clearly a juxtamedullary nephron. Goes quite deep into the medulla. Okay, so when we zoom back out, there are our kidneys. Here's the anterior view, the posterior view. You can see our hilum here. We see our ureter. There's our left ureter. There's our right ureter. And there, the kidneys are going to have this covering of the capsule. So if we follow the ureters, we will then see our so this is the male urinary system, which is going to meet up with the male reproductive system. And right here, we can see where that ureter, so now we're on the posterior side, right? So the ureters are going to enter the urinary bladder on the posterior side. And so here is our whole urinary bladder. And since we're going to have to look at this for reproductive anyway, it might be a good point to point out that you've got your vas deferens or your ductus deferens kind of draping over posteriorly and goes a little bit more inferiorly and meets up with your seminal vesicle. So this is the ampulla, the terminal portion. and it meets up with the seminal vesicle, one of our accessory organs, here at the ejaculatory duct. And if we take off some of these layers, we will be able to see how the urinary bladder drains into the urethra, and the first part of the urethra in a male being the prostatic urethra. And then this short portion that will be our membranous urethra. Oh, let me take off a couple layers. Okay, much better. So now we can see as it goes past the prostate gland, we'll have our prostatic urethra, and then our membranous urethra, and then our spongy urethra. And the spongy urethra is going to start from all the way here and run through the entire corpus spongiosum. Look at this long urethra. So we're going to follow this all the way down to here, and then urine will exit through the external urethral orifice. Okay, so let's take a look at what that looks like in the female. And so here we are with the female system. So assuming that we've got our kidneys here, we'll come down the ureters. And notice how this is the anterior view. And so to really get a good look at how the ureters kind of run a little bit posteriorly, we'll have to go look at a sagittal view. But you see we wrap around posteriorly to the ovaries and then back a little bit anteriorly, antero-inferiorly to the urinary bladder. So it's important to note the relative position of where this bladder is to the uterus. And so when we go back to the anterior view, you can see here this urinary bladder. When the bladder is not completely full, you'll see these folds called rugae, right? Just like we saw in the stomach to help it distend. So here are the little ureters openings. And then this area of the bladder called the trigon. And then from the bladder, you'll go straight to the female urethra. So you see how much shorter the female urethra is compared to the much longer male urethra. Okay. And now let's talk about how urine gets produced in the urinary system. And to discuss the urine flow, we're actually going to start in the kidneys and work our way to the urinary tract. But before we start our discussion of how urine gets produced, we have to talk about the three major functions that are going to get us there. So the first is we're going to take that blood at the glomerular capillaries, and as it goes from afferent arteriole to the glomerular capillaries and out the efferent arterioles, we are going to undergo filtration. And then as you go from so this is going to be where we go from our glomerular capillaries to the capsular space. And what's going to aid in that function is going to be our filtration membrane. So if you look on page 262 and look at bullet point number 11, you will have to know the structures of the filtration membrane and the components that your filtrate has to pass through. Also, don't forget, while it's within the glomerular capillaries, we call it blood, but then the moment it leaves the capillaries and through the filtration membrane and out into the capsular space, we're not going to call that blood anymore. We're not going to call that urine. We're going to call that filtrate. Okay? So our parts of the filtration membrane, as we go from the glomerulus to the capsular space, we're going to go through the capillary endothelium, of the glomerular capillaries, and then we'll go through the basal lamina, and then we'll get to the visceral layer of our glomerular capsule, which again is going to be composed of our podocytes and their many legs. And from the filtration membrane, we will get to the capsular space. The second function is going to be reabsorption. So while we will filter quite a bit of the blood, lots of things are going to filter out into the capsular space that we actually want to keep, like nutrients and water. And so we're going to want to reabsorb a lot of that. And last but not least, secretion. And so let's take a look at which parts of the nephron are responsible for doing what in order. So first we're going to start at our nephron, which is going to be composed of our renal corpuscle. And this is where we're going to see function number 1. And then we're going to get to our proximal tubule, where we'll see both functions 2 and 3. Then we'll see our descending nephron loop and our ascending nephron loop, where we will only see function number 2, reabsorption. And finally, the last portion of our nephron, our distal tubule, which is where we're going to see functions 2 and 3 again. Okay, so as it goes from the capsular space of the corpuscle through the distal tubule, we're going to call it filtrate the whole time. It's not quite blood, and it's not quite urine. But then we're going to take that and we're going to collect them in multiple collecting tubules. And multiple collecting tubules will then take that to multiple collecting ducts. And the collecting ducts are going to go through each medullary pyramid and will drain into each renal papilla. Now, after we leave the nephron, we can actually now call this urine, because after it leaves the distal tubule, we're actually not going to do anything with the filtrate anymore as far as reabsorption, secretion, or modifying the contents further. And so we can call it urine from here on. So then the papilla will drain into your minor calyx, and multiple minor calyces may drain into the major calyx. But remember, it's possible for it to go straight here. But each major calyx will then get to the renal pelvis, which will then exit through the left and right ureters. Okay? So everything up until the ureters is going to be all a part of the kidney. I want to emphasize this is not yet a part of our urinary tract, right? These are everything from renal corpuscle to our renal pelvis. These are components of the kidney itself. And so while about 10% of our blood volume that comes into the kidney will be filtered, so function number 1, about a majority of that fluid is going to need to be reabsorbed. So let's take a look at what parts of our functional unit are participating in each function. And to do that, we're going to need to start at the filtration membrane. So this would be a good time to pause the video and write these notes down if you need to take your notes. And so the filtration membrane as you go from blood to filtrate, those filtration slits are going to be very, very, very thin. And so there's only going to be a certain number of things that are going to be able to pass through those filtration slits. So what exactly can squeeze through? Well, any of our formed elements and a majority of our proteins and platelets even are far too big to pass through the slits. And so what exactly is going to leave? Water molecules. And one thing that a lot of people don't realize when they're getting caught up in the details is glucose is getting filtered. Okay, so I don't want you to ever miss that. Glucose is going to be filtered, but there's going to be a reason why we don't see it in the urine. Okay, what else are we going to see? We're going to see electrolytes. Those are very tiny, very small amount of small proteins. And the subunits of those proteins, amino acids. And we'll also see nitrogenous wastes, which include things like urea, ammonium ions, creatinine, or uric acid. So all of this is just function number 1 that happens as it goes from glomerulus to the capsular space, this filtration. So then, now let's take a look at where it goes next, at the PT, the proximal tubule. So the proximal tubule, this is where we're going to reabsorb a majority of our very, very important things that you want to keep. So what sort of things do you think the body might need, right? Well, we're definitely going to need glucose, right? We need that sugar. We need it so that we can break it down and harvest energy. And so 100%, if there's one thing you remember about the proximal tubule, I want you to remember that 100% of all of our glucose, if our kidneys are functioning properly, 100% of all of our nutrients such as glucose should be reabsorbed at the proximal tubule. Now we say reabsorbed because, and not absorbed, because where does absorption occur? Well, absorption is what we're taking in from the lumen the first time, right? So we saw that in the digestive system with absorbing some of those nutrients from the lumen of the digestive tract into the blood. And since we filtered some of those nutrients from the blood to the filtrate, we are now reabsorbing. So there's a very, very key difference between the terms that we use when we say absorption and reabsorption. What other things are going to be reabsorbed here? Well, we'll take in about 65% of our electrolytes, such as sodium, chloride, potassium. We'll take in 65% of our water ions. And also very, very important, this is where we're going to reabsorb bicarbonate ions, which you can sort of think of as the equivalent to our base. So this is function number 2, that reabsorption. But also, in the proximal tubule, we're also going to see our function number 3, secretion. And so what are we secreting? So what does secretion mean? So with filtration, we went from the glomerular capillaries to the capsular spaces, filtrate. So reabsorption is going from the filtrate back into the blood. And with secretion, we're going from the blood back out into the filtrate to be eliminated from the body. And so the things that will get secreted here include our other nitrogenous waste products. This is where you're going to see a lot of times the secretion of drugs like penicillin or morphine, uric acid, ammonium ions, some more of that creatinine, some urea, and H+ ions. And like we said, in the nephron loop, you're only going to see reabsorption in both parts. So if we look at the descending nephron loop, which I'm just going to abbreviate as DNL for right now. That's not an actual abbreviation. I'm just trying to not write everything out to conserve some space. We're going to see function number 2 with reabsorption. And in the descending part, we're just going to be reabsorbing water. And in the ascending part, when we come back up, in the ascending part of our nephron loop, again, we're going to see function number 2, but this time we're more concerned with salt. In the distal tubule, what are we going to reabsorb? We're going to reabsorb some more water, some salt, and calcium ions. And we will secrete potassium and hydrogen ions. And I actually almost forgot to mention, even though this is not a part of the nephron loop, even though this is not a part of the nephron, even though this is part of the collecting system, we do see a little bit of reabsorption happening here, mostly under the influence of hormones, especially that help regulate our fluid and electrolyte balance in the body. And so things like anti-diuretic hormone will enable our collecting ducts to reabsorb things like water. And we'll also take in salt, and this will be the last place we will reabsorb any bicarb